Okay, so now what I'd like to talk about is I'd like to talk about each of these steps involved in going from action potential in the motor neuron all the way to muscle contraction. I'd like to talk about them in depth. And so the first thing that I'd like to talk about here is neural control and excitation. And what that means is that means we are going to be talking right at this level. Here's the motor neuron. And how does an action potential in the motor neuron trigger an action potential in the muscle cell? Okay. What I'm going to do is I'm first going to go through this in PowerPoint slides that match what's in your textbook, and then I'm going to show you an active learning exercise where you can print things out and practice drawing and moving the pieces yourself at home, in case that is also helpful for you to do. Okay, so what we're going to do is we're going to go close up. You can kind of see it right here. Okay, and so what we're seeing in this image is that right here is the boundary of the motor neuron. Excuse me. Over here is the boundary of the muscle cell. It's the plasma membrane or sarcolemma. Just like what you saw with synapses before, there is a gap between these two cells. Okay, and that is going to be called the synaptic cleft. Now this part of the sarcolemma that is right across from this motor neuron is called the motor end plate. So again, the motor end plate is part of the sarcolemma that is directly across from the motor neuron. Now, just like we saw with synapses, we have vesicles in this motor neuron that are packed with neurotransmitter. And this neurotransmitter is called acetylcholine, ACH for short. And to write that out for you, ACH stands for acetylcholine. Okay, and that is a type of neurotransmitter. This is the neurotransmitter that is always going to be used to stimulate muscles. Okay. So again, motor end plate, synaptic cleft, vesicles with neurotransmitter, and this whole structure is called the neuromuscular junction. Junction means the coming together of two things, and what do we have coming together? A neuron and a muscle. So neuromuscular junction. Okay. Now, on the motor end plate side is going to be acetylcholine receptors. Okay. Now this is before contraction, so this is at rest. And we're going to talk about this molecule, acetylcholine esterase, that is found in the cleft in a little bit. Okay, so the way we start is we have an action potential that comes down the motor neuron. That is going to cause acetylcholine to be released into the cleft. It diffuses across the cleft from the motor neuron over to these ACH receptors. When these receptors open, they allow sodium to come into the muscle cell. And that will then trigger an action potential. That's what this red is showing here. That will trigger an action potential along the muscle cell membrane, along the sarcolemma. Okay. So let's go that far with the active learning pieces, and let's put those together. Okay, so we have the pieces cut out here from the active learning exercise that is posted online. And so some parts you're going to draw and some parts you're going to place onto your drawing. So the first thing we're going to draw is we want to draw the boundary. Okay, this right here is our motor neuron. Okay, so this is our motor neuron, and our motor neuron is going to be filled with vesicles. Okay, and that's what we're drawing here. And those vesicles have our neurotransmitter, acetylcholine, in it. Okay, so we're going to take acetylcholine, we're going to place acetylcholine in our vesicles. Again, this is before contraction has occurred. The next thing we're going to draw is we're going to draw the motor end plate. Okay, so inside here, this is going to be muscle cell. Okay, and that's sarcolemma of the muscle cell. So muscle cell sarcolemma. So we want to place a few things in the correct location. First, we have our ACH receptor. So this here is showing ACH receptor. We place that along the sarcolemma. Another couple of things that I'll have you go ahead and place. So I do want you to go ahead and place a sodium pump along the sarcolemma. Just like what we saw with the neuron, we do have sodium in the cleft. Okay, so this region across here is that cleft. So technically sodium is outside of the cell in the cleft. I want to go ahead and draw a couple other things in this image at this time. One is we're going to have the sarcoplasmic reticulum. Okay, so this is, we are now inside, I can't quite see that. So this here is inside the sarcoplasmic reticulum. And what we have that's very important inside of there is we have calcium. So calcium stores are there. We're going to have a couple of important things. We're going to have a calcium channel. It's closed, and it's on the sarcoplasmic reticulum membrane. We're also going to have a calcium pump in the same membrane. Finally, the last thing that I do want to go ahead and put in this picture is the sarcoplasm, okay, and the sarcomere, okay. So in here, is sarcoplasm. And so the sarcomere, all of these myofibrils are located in the sarcoplasm. Okay, so just to recap, in here is the motor neuron. It's got vesicles with acetylcholine in it. This line here is representing the muscle cell sarcolemma, specifically the motor end plate. Here is a gap called the synaptic cleft. Over here is the boundary of the sarcoplasmic reticulum. And here is sarcoplasm, which is cytoplasm of the muscle cell, and that's where we find our sarcomeres. We've got calcium stored inside the sarcoplasmic reticulum. We've got sodium in the cleft. We've got a closed acetylcholine receptor along the motor end plate, a sodium pump along the motor end plate, a closed calcium channel on the sarcoplasmic reticulum, and a calcium pump along the sarcoplasmic reticulum. One more thing that we now need to put into the picture that we have not talked about yet is troponin and tropomyosin, the wavy things that are in the sarcomere file. Those cover up actin, and they prevent actin and myosin from interacting with each other. Okay? So if troponin and tropomyosin are present, myosin heads cannot bind to actin and pull them closer. Okay, so let's start now with what happens to give us a muscle contraction. And let's start with an action potential in the motor neuron. So the first thing that happens, action potential, okay? Action potential, okay? It's going to travel down that motor neuron membrane. When it does, these vesicles will come here and they will fuse with the plasma membrane and release acetylcholine into the synaptic cleft. Acetylcholine will diffuse across and physically bind to the acetylcholine receptor. Once it does, this receptor opens. After it opens, sodium comes through that receptor into the muscle cell. That triggers action potential along the muscle cell membrane. It will go deep down the T-tubules. And note that there is a location where T-tubule and sarcoplasmic reticulum are very close to one another. Remember that action potential is a change in voltage, and it turns out this calcium channel right here is sensitive to voltage. So as soon as the action potential hits this triad region, this calcium channel will open. And when it does, calcium rushes into the sarcoplasm. What calcium then does is it will come over to troponin and tropomyosin and pull it off of actin. So calcium comes, pulls troponin and tropomyosin away from actin. Okay? And as it does that, myosin can now bind to actin and pull it tight, and you get a contraction. As long as calcium is present in the sarcoplasm, this muscle will contract. Now, we want to contract, but we also want to relax. So how do we get this thing to turn off? Remember, as long as calcium is in the sarcoplasm, the muscle will contract. So it turns out we have one player that we haven't placed on the board yet. And that player is one that I've made look a little bit like a Pac-Man. It's called ACHE, or acetylcholine esterase. And it lives in the cleft. What it does is it comes over to acetylcholine and it chews it up. So as it comes along and chews up acetylcholine, there's nothing bound to the receptor. There's no more acetylcholine bound to the receptor. So that receptor will close. And guess what's there to take the sodium back? A pump. So sodium is then pumped back into the cleft. Now, since sodium is no longer in the cell, action potential is no longer happening along the muscle cell. So this calcium channel is no longer triggered to be open. So it will close. And then guess what's there? There's a calcium pump. And that calcium pump will take the calcium and it will pump it back into the sarcoplasmic reticulum. Now notice, calcium is no longer bound to troponin or tropomyosin. So troponin, tropomyosin will hop back onto actin, covering it up, and it will prevent actin and myosin from interacting. And when they can't interact, this sarcomere now lengthens, causing the muscle to relax. Okay, so these printouts are posted online, and you can print them out yourself. I would practice drawing, moving these pieces, explaining it, until you can do all of it by memory without having to look. One other piece that I'd like to add a few more details onto is the nature of the interaction between myosin and actin. So what we're looking at here is one myosin filament, one thick filament. Here's one of the heads poking out. Here's another thick filament. Here's a couple of heads poking out. What this is showing here is this is showing actin. You see the big circles are actin, and these thinner things are troponin and tropomyosin, and notice that calcium is there, so it's pulling it away from actin enough that this head of myosin can now physically bind to actin. That's called forming a cross bridge. What I want you to look at is I want you to look at ADP, phosphate, and ATP. Remember from chapter 2, ATP is our currency of energy in the cell. So if we look at this head, this myosin head, it's got ADP and a phosphate. Notice here it is kicked out. This is the next step. It is kicked out the ADP and the phosphate. It no longer is physically associated with it. And that allows this head to come up to actin and then pull on it. That's called a power stroke. And that's how you get those filaments to slide past each other. Notice this one is doing the same thing at the same time. Once it's finished pulling on it, okay, ATP now jumps into this head. Once ATP does that, it is broken into ADP and phosphate, and that allows this head to recock itself and then bind, and it will now pull on the actin. And so that is how this cycle goes. So the important thing to note here is that the name of a myosin head pulling on actin is a power stroke. The other thing to notice here that is important is that every time the myosin head recocks itself, resets itself, you burn 1 ATP. There are thousands and thousands of myosin heads in every single muscle in your body. And every time one of them pulls, it requires 1 ATP. So you can imagine that this is the molecular basis for why muscle cells require so much energy. Okay, and so what I'd like to point you out to in your notes that we have discussed is, if you go here, here's a place, another place for you to draw the neuromuscular junction before a contraction occurs. These are all the things to put in the correct places. And then see, given what we've talked about, can you list out the steps that take you from action potential to contraction? And then, can you list out the steps that take you from contraction to relaxation? Okay. And then an even bigger thing to test your understanding is here, where our A&P character Manny accidentally ingests a poison, and it affects this channel, and see if you can predict what will happen. And then Manny gets electrocuted by grabbing an electrical wire. His hands contract around the wire and he's unable to let go. And so see if you can understand why. Okay, so you have that information. See if you can figure that out. And contact your instructor if you need help with that.